Welcome to our senior secondary biology. In this video, we're going to take a look at plant nutrition part 1. The topic is going to be delivered into two videos. This is part 1 and there will be part 2. Let's get started. So when we introduced nutrition, we talked about different types of nutrition such as saprophytic nutrition, autotrophic nutrition, and here we're going to start autotrophic nutrition. In short, this type of nutrition that we're going to take a look at, which is plant nutrition, is an example of autotrophic nutrition. We're going to start with the most important process in the universe when it comes to energy manufacturing or food making, which is photosynthesis, and is defined as the process by which green plants manufacture glucose from carbon dioxide and water in the presence of light energy absorbed by chlorophyll. So we have glucose, which is food being made, and we have carbon dioxide and water reacting in the presence of light energy, which is absorbed by chlorophyll. So in this process, oxygen is produced as a byproduct. The process takes place in leaves. So even in some leaves which have different colors, it's not all the parts where photosynthesis takes place. Photosynthesis usually takes place on green parts of the leaf because the green parts of the leaf are the ones with chlorophyll. This chlorophyll actually is the one that gives the green color to plants. This is the word equation which summarizes the process of photosynthesis. We have carbon dioxide reacting with water to form glucose and oxygen. Glucose is the main product, oxygen is just a byproduct. And this happens in the presence of light energy and also chlorophyll. This chlorophyll absorbs this light energy. Then this light energy is going to do some miracles here so that the reaction is possible. We're going to start this in details. So the oxygen that is released, the oxygen is released out of the plant. Well, some of it is used for respiration. So this oxygen, part of it is taken outside the plant for animals, of course, to breathe in, and part of it is used in the process of respiration, which we defined when we started learning biology. So the entire process of photosynthesis is made of two reactions or two stages. The first stage is called the light stage or light reaction, and the other one is called the dark stage or dark reaction. The following is what happens in the light reaction. There is a process which is called photolysis, and this is photolysis. So in photolysis, we have water being broken into two constituent elements, which are oxygen and hydrogen. Water is made of oxygen and hydrogen, so it is going to be broken into oxygen and hydrogen in the process of photolysis. So what breaks water apart? It's the sunlight or light from the sun. So the light from the sun, once it is absorbed by the chlorophyll, it ends up breaking water into oxygen and hydrogen. So this process is what is called photolysis. By the way, photosynthesis is made of two words, photo and synthesis. Photo means light and synthesis means making. So photosynthesis is making something using sunlight. Then photolysis is made of photo and lysis. Anyway, in this case, we can take it as breaking apart or splitting. So water is broken into two parts using the energy from the sun. So once water is broken into oxygen and hydrogen, what happens? Oxygen, as we discussed, diffuses out of the plant for animals, of course, to breathe in, and part of it is used for respiration. What about hydrogen? So hydrogen is the one that proceeds to the next stage of photosynthesis, which is the dark reaction. So in summary, oxygen diffuses into the atmosphere while hydrogen proceeds into the dark stage. So what happens in the dark stage? So in the dark stage, what happens is simple. The hydrogen that comes from the light stage combines with carbon dioxide, forming glucose. There is no light involved in the dark reaction. So this is what happens in the dark reaction. And the word equation is hydrogen combining with carbon dioxide to form glucose. So we're going to do an experiment to determine if photosynthesis has taken place. How can we know that photosynthesis has occurred? So if we have a leaf, the only way we can know if photosynthesis has occurred is to test if the products of photosynthesis are there. There are two products of photosynthesis. There is oxygen and there is glucose. Oxygen usually diffuses out of the plant, so we can't detect oxygen usually, but we can detect the presence of glucose. Remember, glucose is mainly stored as starch. So if we can do a test for starch on a leaf, then we can be sure that photosynthesis took place in that leaf, because there is starch which comes from photosynthesis. So we're going to do a couple of steps that are going to help us um, identify the presence of starch. So the first process, or the first step, is you boil the leaf that you want to test in water. So get your leaf, then you put it in water, you boil it. So why do we boil it? We want to kill protoplasm. Why do we want to kill protoplasm? Remember, the cell membrane chooses what enters the cell and what leaves the cell. So if we want to test the presence of starch which is inside the cell, we need to add iodine inside the cell. But the cell membrane might not allow iodine to go inside the cell. Therefore, we'll be unable to reach the starch which is inside the cell. So what we do is we have to kill the cell membrane to destroy it by boiling the leaf. So once the leaf is boiled, iodine will be able to penetrate the leaf. But before we can even add iodine to the leaf, there is something that we should do, which is the step 2 of this experiment is to put your leaf into boiled alcohol. So you boil the leaf in alcohol. Why? So the chlorophyll, we want to remove the chlorophyll. Why do we want to remove the chlorophyll? When we are testing for starch, we expect the iodine solution to turn blue-black if there is starch. But if there is this color, it might interfere with the results of the experiment. So we need to make sure that there is no any color on this leaf for us to test. So we boil the leaf in alcohol so that we remove chlorophyll so that it does not interfere with the color changes. One thing to note is when boiled with alcohol, be careful. You have to use a water bath. Why? Because alcohol is flammable. So to avoid that, we use a water bath so that there is no fire. The third stage is to put your leaf, or to soak your leaf in warm water. So get your leaf which has no chlorophyll. Now as you can see, it's not green, it has no chlorophyll. So you soak it in warm water. Why? Alcohol makes the leaf brittle, it can break. So to make the leaf smooth, you put it in warm water, soak it in warm water. Finally, we have our leaf which is not brittle, which has no cell membrane so the iodine can penetrate, and which has no chlorophyll, which can interfere with the results of the experiment. We're going to add iodine solution to the leaf so that we test for starch. So when we do this, a drop of iodine solution is going to penetrate the inside of the leaf, and it's going to, of course, expand like this. And if there is starch, then this solution is going to turn blue-black. Therefore, this proves that photosynthesis has taken place. So what is needed for photosynthesis to take place? So what is needed is what we have already discussed, and that is carbon dioxide, which is a reactant, water, which is a reactant, and two factors, one of them being the sunlight and chlorophyll, which implies that in the darkness, photosynthesis cannot occur. If there's no sunlight, photosynthesis cannot occur. It also implies that the parts of the plant which are not green, photosynthesis cannot occur, because chlorophyll is found on green parts of the plant. So we're going to do a few experiments, maybe 3, so that we find out if carbon dioxide is really important for photosynthesis, and if sunlight is really important for photosynthesis, and also chlorophyll. We're not going to do one for water. It's not the main concern in the syllabus. So we're going to start with carbon dioxide. So the first step is you discharge a well-potted plant. So what is to discharge? To discharge is to remove starch from the leaf. Why do we want to remove starch from the leaf? We want to remove starch from the leaf so that when we start doing our experiment to test if photosynthesis has taken place, we cannot claim that photosynthesis has taken place using the starch that was produced some time ago. So in short, the idea is we're going to take a plant, we remove all the starch in it, then we allow it to undergo the process of photosynthesis, then we test again if there is starch. If there's going to be starch again when there was no starch, then we can be sure that photosynthesis has taken place. So to destarch a well-potted plant is just you put it in the darkness for 4 to 6 hours so that all the plant that is in the all the starch that is in that plant is used up. Once that is done, you can take your plant and put it in sunlight so that photosynthesis takes place. So we're testing for the presence of carbon dioxide. So we need two things. We need one where we have carbon dioxide and a control experiment where we don't have carbon dioxide. So what I'm trying to say is, we set up the plant as below. So you have this. So you select two leaves, which you of course should have enough chlorophyll. As you can see, they're all green. Once you do that, you put some solutions here. So these solutions are very important and you should know their importance. The first one is sodium hydrogen carbonate or distilled water. So these two make sure that there is enough carbon dioxide here. But the other one on the other side is sodium hydroxide, which absorbs all the carbon dioxide. So I want to see if we're going to find if we're going to test for starch here, if we're going to have a positive test for starch here or here. So in short, the sodium hydrogen carbonate or distilled water makes sure there is enough carbon dioxide, well this other one absorbs all the carbon dioxide. So we've got two situations here, one with carbon dioxide and the other without carbon dioxide. We're going to see what photosynthesis is going to take place. So after you leave this in the sunlight for hours, you can test your leaf for starch by adding iodine solution to each of these leaves. And when you do that, what you are going to discover is the leaf on the left, which this one with enough carbon dioxide, will test positive for the presence of starch, while this one is not going to test positive for the presence of. The second experiment is to show that light is necessary for photosynthesis. So again, you discharge a well-potted plant, then you set up the plant as below. So in this case, we're going to get a leaf, then we're going to use an aluminum foil to cover parts of the plant. And of course, we're going to expose some parts here and in between here. And we can use paper clips to hold this cover. So the idea is, as the sunlight comes, this part here won't be reached by the sunlight. So don't expect photosynthesis to take place, since we're claiming that sunlight is important for photosynthesis. So once we do that, at the end, when we're done with the experiment, of course, you can even draw this and label the parts so that you are able to keep track of which parts are exposed and which parts are not exposed, because you need to remove this aluminum foil for you to put your iodine. So when you do that, we're going to discover that the exposed parts will test positive for the presence of starch, because sunlight is able to reach the exposed. Finally, we're going to test if chlorophyll is necessary for photosynthesis. In this case, we're going to use a special type of leaf called a variegated leaf. So we're going to discharge a well-potted plant with variegated leaves. What is a variegated leaf? A variegated leaf is a leaf with more than one type of eye color, like this one. This has green here and it has got white. So this is a very good example of a variegated leaf. So once we have a variegated leaf, we expect here where it's green, where there's chlorophyll, to undergo photosynthesis. Well, here where it's white, not to undergo photosynthesis. So we're going to place the potted plant in sunlight for 4 to 6 hours. And then of course we can just draw this so that you are able to trace which part is white and which part is green, because when you apply iodine, you won't be able to tell which part is green and which part is white. So you can label the parts. Maybe this one you call in your drawing, you can call it part A and the other one part B, so that you are able to track. So once that is done, you're going to discover that the green parts will test positive for the presence of starch, while the white part won't test positive, meaning that photosynthesis has taken place here only, not here. Finally, we're going to take a look at how we measure the rate of photosynthesis. How can we tell that the photosynthesis is high at this point and is not high at this point? So we're going to set up an experiment as below. So what we have here, we have got a pondweed, which is an aquatic plant. An aquatic plant is any plant that is usually underwater, so is able to survive underwater. So, and we're going to have all these experiments set up. We're going to have a test tube here, and of course water, inverted funnel. So what we expect is As photosynthesis is taking place, this plant is going to be releasing oxygen, since oxygen is a byproduct of photosynthesis. This oxygen is going to be in form of these air bubbles, which of course will try to go up the water to this part here. So how do we measure the rate of photosynthesis? The number of bubbles being produced in a specified period of time will tell you the rate of photosynthesis. If a lot of bubbles are being produced, it means the rate of photosynthesis is high, and if a lower number of bubbles are being produced, then the rate of photosynthesis is low. So this is how to measure the rate of photosynthesis. This marks the end of this topic. Hope you have liked it, and I hope you have learned a thing or two. Happy solving.